Hello everybody, welcome to lecture 2 of our computer vision topic. We're going to be talking about chapter 2 of our book Digital Image Processing by Gonzalez and Woods. This lecture is divided into 5 parts. We're going to be talking briefly about the elements of visual perception. Then we'll discuss a little bit about visual illusions, go into image sensing and acquisition, how to represent digital images, and the most important part of this lecture, which I ask you to concentrate on, is the fundamental mathematical tools that we're going to be using throughout this topic. Let's start then with lecture 2.1, elements of visual perception. There are obviously in the animal kingdom, there are many varieties of visual perception. Various animals have distinct ways of seeing things, of perceiving things, but as it's a course on computer vision, as we discussed in the first lecture of the topic, the goal of computer vision is to try to make machines understand the world or describe the world in the way that we describe, and we describe the world that is seen by our eyes. So we're going to concentrate on elements of human visual perception. It's mostly based on the human eye. The human eye is nearly a sphere. It is enclosed by 3 membranes: the cornea and this sclera outer cover. This is the sclera. The cornea is here. Let me change here to a pointer. All right, this is the sclera, and this is the cornea. And the choroid, which is this middle membrane here, that has a ciliary body, and the iris, which has a very important task, which is to contract. It contracts and expands in order to control the amount of light that's captured by the lens. The retina is where the image is formed. This is the 3rd membrane, that's the innermost membrane. And the lens, which this part here, is composed by a set of concentric layers of fibrous cells, basically act as a glass lens, as a camera lens that retracts light to be focused on the retina. And it's interesting to make the analogy about a normal old school analogic camera. Then we're going to understand better the functioning of all of these membranes. And there is this wonderful documentary about how the human eye works by the BBC. I'm going to just pass to you now in this lecture a couple of minutes about of this documentary. And I remember that I've seen this documentary in the 80s, and it still turns me on every time I watch it, because it has all of the elements that we need to know for understanding, for the purpose of this topic, of course, for understanding visual perception. So I'll pass it in a minute.
And even some lizards see much better than we. Nevertheless, our eyes are truly wonderful mechanisms. The human eye, like a miniature camera, has an iris to regulate the amount of light coming in and a lens. The shape of the lens of the eye can be changed by tiny muscles to suit the distance at which it has to work. The eye has a dark chamber and a sensitive layer called the retina, a counterpart of the film in the camera. And back of the retina is a layer of black tissue, which, like the black paint inside the camera, prevents reflections which would blur the image. For an object to be seen sharply, The light rays coming from it to the eye must be focused so that an image falls exactly on the retina. The retina consists largely of nerve fibers. On its back surface are the so-called visual cells, the rod visual cells and the cone visual cells. A light ray which strikes the transparent retina passes through it to the outer parts of the visual cells. Chemical changes caused by the light start a nervous current back through the retina along its inner surface and out through the optic nerve to the brain. Here, a sensation of light is aroused. The rod visual cells are hooked up in groups to one optic nerve fiber so that they cooperate in setting up a nervous current. For this reason, they can cause a current flow in very dim light. The cones, however, are not usually hooked up together, so the light must be brighter before the cones can function. At night, therefore, only the rods can function. But because a whole patch of rods is connected with each brain cell, each point is registered as a blur, and things are not seen sharply. When the light is brighter, the cones come into play. Because each cone has a private wire to its own brain cell, they provide the sharp details of the picture. But the cones, the organs of sharp, bright light vision, do more. They give us the fascinating world of color. In each cone are 3 chemical substances. Each substance is sensitive only to a particular group of colors. Thus, one is most sensitive to red, one to yellow, and one to blue. When a particular color of light strikes the cone, the corresponding substance breaks down, and a corresponding nervous current flows to the brain. When a different color, yellow for example, strikes the cone, the yellow substance sends a nervous current to the brain. And when blue light strikes the cone, a still different nervous current informs the brain of the color. However, the cones do even more, for they can create a corresponding nervous current for any and all colors by a combination of their sensitive chemical substances. When all...
Okay, so after you've learned a little bit about image processing, digital image processing and computer vision, I'll ask you to return to this video. Then you are going to see how good it is. That it's still what we're trying to do in computer vision and image processing is exactly what the brain cells, what the visual cells are doing. What we're trying to do, we don't do as well as the human cells, as well as Mother Nature, but we, as engineers in computer science, we try our best. Okay, so let's discuss a little bit further this. So we've seen that that basically the visual cells are rods and cones, and this is the distribution of rods. So rods are largely distributed throughout the retina, but not at the center of the eye, of the visual field, which is the fovea, where that's the place where the rods are more abundant. The rods, as you've seen in the video, they are connected to, each rod is connected to a single neural cell, neuron, and there's the rods that are responsible for perceiving details and perceiving color, whereas the cones are more responsible for perceiving the scenes and for perceiving the environment under dim light. And you see that there is a point here where there is no rods or no cones. This is the blind spot. This is the blind spot. We really don't see anything at the blind spot. You don't see a black hole in the middle of your eye because your brain is covering it up with information. It's making a kind of interpolation of pixel levels around it. Okay, so the retina, as we're seeing, picks up the light from any object, and basically is the image is formed on the retina. Patterned vision is afforded by discrete light receptors, which are the cone, primarily located in the fovea here. They are highly sensitive to color, specialized in fine details in bright scenes, and that's why they are connected to one nerve cell per cone. Whereas the rods are largely distributed around the retina. There are several rods are connected to single cells, and they capture the overall image of the field of view. They are not involved in color vision, and they are very sensitive to low levels of illumination, whereas the cones are they are not sensitive at all to low levels of illumination. Like in a photographic camera, just to pin down what we've just seen in that BBC documentary, the lens has a fixed focal length in a photographic camera, so focusing at various distances, you vary the lens, or imaging the plane distance, you change the distance of the lens to the focal length. We actually don't have a we don't change the distance of our lens. On the other hand, the human eye changes the focus by changing the shape of the lens. The shape of the lens. We don't change the focal distance of the lens by changing the location of the lens on our eye. That would be very good if we had it, but we actually change the shape of the lens by these fibers called ciliary body. And perception, human perception, visual perception, is just double quoting, right? The relative excitation of light receptors, what they are doing, they are transforming radiant energy into electrical impulses that are decoded by the brain. And one thing that we have to take into account, that's a very important thing and very relevant when you are developing computer vision algorithms or image processing algorithms, is that our eye is adaptable. We see a wide range of light intensity, basically 10 to the power of 10, because that's our range of perception. But we don't see everything at the same time, which meaning that if we are in under the bright sunlight and we go inside of a cinema, I think everybody had this sensation. Once you get inside of the cinema, you don't see anything. It's just black, pitch black. But if you stay a little while in the cinema, your eye adapts. That's what's called brightness adaptation. So we have an adaptation level. For an adaptation level BA, you have a different perception of very dark to very bright. So under this point, this point is you are now rather bright scene. The very dark is a point here. Let's say a point X. Oops, that's a point X here, and the very bright is a point Y here. If you are in a dark room down here, your perception of what is very dark will be another point here that's lower than this initial BP here. So your notion of pitch black and very bright changes with respect to the environment. We are going to, as I said, we're going to return to these points throughout this topic, but we're going to see how this influences our visual perception when we talk about visual illusions in the next section of this lecture. And if you want to learn more about the visual perception, human visual perception, please read section 2.1 of our book. And I'll be back soon with talking about this exciting topic, which is which is about visual illusions. Just love this topic. So hold your breath. I'll see you soon.